overview of Bgee. What is Bgee? What are we doing, and what can you do with it? So first, Bgee is the work of a team. Obviously, we're two of us presenting here. The photo I had in the previous slide was from last year. This is from this year. So we are a team of 10 people, curators, developers, PhD students, and PIs. And we can be contacted by email, but also on social media. You see here our Blue Sky, Mastodon, and LinkedIn. So don't hesitate to reach out to us. We try to answer rapidly, whatever the way you contact us. The goal of Bgee is the intersection of two questions: understanding gene expression and helping biologist users. So what we want to do is to make gene expression useful to biologists. Gene expression is very interesting, very important, but also very complex. There's a lot of data of different types in different circumstances annotated to complicate that. It comes from complicated anatomy, cell types, it uses different technologies. We want to make all this as usable as possible to do the boring work as well as possible behind the scenes so that you can advance on the science. So I will first do a little demo of Bgee. So take you through Bgee. So I have to redo the sharing. Wait, how do I recover? Okay, so this is the homepage of Bgee. Do you all see it? Frédéric, do you see the website? Okay, and so the first thing you see is this button, gene search, and if you type here, you can search for your favorite gene. So I'm going to click insulin here, but you can type anything, and it gives you this gene in different species. So here, for example, the insulin of human, and then what you find is a description of the gene, where it's expressed in a graphical way and where it's expressed in text. And here you see the different structures, a score which says how highly it's expressed, false discovery rate of the probability that which gives you the confidence we have this gene is actually expressed here, and the sources. And you see here we have different sources which are integrated together: RNA-Seq, single cell, Affymetrix, in situ ESTs. Okay. And if you look, we have a diversity of species. So if you unfold here, you can have different information. So here I have the species, and you see that we have 52 species. All the species we have are animals. And for all these animals, we curate and integrate these different gene expression data. You can also, and we will see over the day what these different buttons lead to, what they show you. So this is the general overview of Bgee. And if I go to WooClap, and sorry, and if I go to the, so I should say in Bgee we only have animals, okay? So now I'm going to put you on new WooClap to see that you've been following. So do we have the data for the following species in Bgee? Hide the results. So the WooClap link is always the same one. The questions change, but it's always the same link. Just you will find on your browser the questions renewing themselves, okay? Five, 4, 3, 2, 1. So do we have, a lot of you notice that we have data for fly Drosophila melanogaster. It is correct. We have all of the animal model organisms. So Drosophila melanogaster fly, C. elegans nematode, zebrafish daniorerio, mouse mus musculus, all these major model organisms of animals, we have them, and we interact with the model organism database. If you work on fly, you know FlyBase, WormBase, MGI, ZFIN. We collaborate with them to have the best data together. Do we have Platypus, we have platypus. Indeed, we try to have data from a wide variety of animals, including non-model species. So that's one of our originalities. We have both the model organism, but also the non-model organism, which allows you to compare expression if you want to compare between species, or if you work on those species to recover the data. Do we have human healthy data? Yes, we do. We have a lot of human data, and importantly, and this was a bit of a trick question, but I didn't tell you before, I wanted to see the answers first. We curate the healthy data, so we only show you healthy data. And so do we have human cancer? No, we do not. Because we curate only healthy wild type data so that you can be guaranteed when you come to Bgee to find a reference of expression data which is under the most standard normal conditions, which can allow you to compare between species what is comparable. You don't want to compare human cancer to a mutant mouse. You want to compare a healthy human to a wild type mouse, and allows you to have a reference for those of you who do biomedical research. If you have data from disease or treatment, you want to know how the gene expression looks like when there's no disease and no treatment, and this is what we provide you. Do we have yeast? Do we have Arabidopsis? No, we only have animal data. As I showed you, we have different types of data in Bgee. So for this, we curate all the different types of data from their raw sources, so the sequence read archive, Geo, ArrayExpress, and so on. We identify valid data sets, so it needs to be a technology we support. Notably, there's a wide variety of single-cell technologies we'll speak about later this morning. Some we support and some not yet. And as we said, we identify that the data sets are healthy wild-type samples. It can be that in the same experiment there are some healthy samples and some disease, or some wild-type and some mutant, and then we will identify and only take the healthy wild-type samples. And then we retrieve all the necessary files, which sometimes can be quite simple, but sometimes quite complex, because you can have files such as the barcode files or the association between barcode and cell population, which are not in the primary database, but in supplementary data or in other places. Then we annotate these samples with the information about the experiment, which allows us to provide you with this information and also allows us to do the right analysis on it. And we annotate two ontologies, which we'll explain a bit more in detail later, but allow us to standardize what they are annotated to, whatever the vocabulary and the terms that the authors used. Then we process all that data so that we can give you those numbers I showed you on the gene page, how highly is a gene expressed, and whether it is expressed actively or not, yes or no. And we integrate all of this into Bgee, into all our different tools that you will see over the morning. Now, the first things I showed you here is curation and annotation. And this is part of what we call more broadly biocuration. Biocuration is a very important effort which is done behind the scenes in different bioinformatics resources. It's one of the things which puts us apart from resources which are not curated, and so I want to explain it a bit.
So before you start, there is a question that is relevant to what you're discussing. How do we choose the species integrated?
Okay, so how we choose the species integrated? A, we restrict to animals. B, we need to have a high quality genome with annotations. C, we need to have sufficient gene expression data so that we can actually show you expression in different organs or different cell types. So if, for example, some small insect, the only gene expression data available is the whole body, we cannot really exploit this. I should add that right now we have 52 species. We have plans to go to 200 in the next year. We have already prepared the curation of that data. It takes a bit of time because we need also to scale up the database and have all the genomes up to date. But that is how we choose the species. In the future, our long-term plan is to have all species which have these conditions, good genome and sufficient expression data in different organs or cell types and animals. So I hope this answers the question. To come back to our curation, so there are two types of databases in biology, uncurated and curated. Uncurated databases, a typical example we have all used is NCBI nucleotides, which we also know as GenBank. it has advantages that it's very complete and very up to date. Someone puts data into GeneBank, submits a sequence, the next day it's there. And anyone who submits sequences to GeneBank, they are all there. So it is very complete and very up to date, but it has a lot of data redundancy. You'll find the same sequences many times, and there's a low organization of knowledge. You cannot be sure to find a gene by its name. You cannot be sure that any details were well put together. Maybe a protein sequence is there without its domains, and so on. The other type of database are curated database. In curated database, curators, people, experts verify all the information. So the data are verified, there's minimal redundancy, the annotations are standardized. So the added value here is that you have organized and reliable knowledge. On the other hand, it might not be as complete because it takes time for humans to do the curation, and so the raw data comes out much faster than the curation does. An example most of you I think would be used to is the Swissprot part of UniProt. So when you go to UniProt, the part which is curated, which has a golden star, was verified by curators. And this is the definition of biocuration by the International Society for Biocuration. Biocuration involves the translation and integration of information relevant to biology into a database or resource. So translation and integration, this translation is very important. The expert curators will take the terms that are used in human way of writing in papers, in the first description of a data set put to a database, and translate it to standardized terms that can be analyzed automatically and relevant to biology and to a database of resource that enables integration of the scientific literature as well as large data sets. Now, what curators do, one of the things we do is to choose the right data sets, but another is to do annotation. Annotation is associated a biological object to a feature. That sounds a bit dry, so I'll give you an example. If you have a gene and it has a gene ontology term, so you have HOX and it's a transcription factor. You associate this gene ontology term to this gene, it's an annotation. And this annotation can be done one of two ways. Uncurated, so you just say, I do a blast, and the first blast hit, I take all its gene ontology terms and put them on my gene. This is uncurated, it's unverified. Or I can read the papers, the experiments done about the gene, find what functions it has described in the literature, and translate these to gene ontology terms. That is curated annotation. And Bgee, why am I saying all this? Because we are a curated database. So all expression data in Bgee is verified, and data which does not fulfill our criteria is excluded. So we do not aim to have all of the data, we aim to have all of the data which fulfills our criteria. Healthy, wild type, protocols which can be processed in a standard way. And then the expression data sets are annotated by manual curation, which means that our curators read all the metadata, read the paper, and when things are unclear, they contact the authors so that we have the best possible information. And the annotations follow standards, and these standards themselves are established by curators working together to make standards which are very careful and standardized. So let's go back to WooClap. Can you think of some examples of curated databases that you know? So here, I suggest that you write directly the name of a curated database you know, and if someone wrote the name of the database you're thinking of, you can upvote it. Yes, thank you, Bgee. But some others. Okay, so let's see what came out of this. So here there is Geo. Geo is not a curated database. GEO is to gene expression data what GenBank is to nucleotide sequence data. People can put data there, and if you write something unclear or not following standards when you submit your data, it will stay like that. If some information is missing, it will stay missing. So there's not a curation. GEO tries to be complete, is a primary database where people can store their data and share it, but it is not curated. The same goes for RNA-Express. These are primary databases of gene expression. UniProt has two sections, SwissProt, which is indeed curated, and Tremble, which is not curated. So UniProt is actually it's a mix of the two. I suppose this is SwissProt. SwissProt is the society which manages luggage in airports, but I don't think that's what you meant. Thayer and Flybase and ZFIN are all model organism databases for respectively Arabidopsis, Drosophila, and zebrafish, Daniorario. They are indeed curated databases with expert curators who check information about knockout phenotypes, about gene expression patterns from in situ hybridization, about different genetic lines, and so on. So these are curated databases. Celosaurus is not a database per se, but it is curated. It is a curated list of cell lines with their descriptions and clarification of the terms. GTech is a very large data set. I hesitate to call it curated, so the people in the GTech consortium have tried to do the best job possible of naming their data sets, but it is their data set. So we don't usually call it a curated database when it's the people who generated the data set who make it available, because these are not independent professional curators. So we have recurated GTech, as we will show you a bit later. ENA is the primary database of nucleotides, the European version of GenBank, so that is not curated. Some of these I don't know, so I don't know what is blood spot. I will look at it later. Thank you. These were interesting examples. So our curation, as I told you, is only to take wild type healthy. So why wild type healthy? First, it's informative on what we call the causal function of the gene. So a gene, you can describe function in different ways. For example, you can say that BRCA1, BRCA1, is involved in breast cancer, but that's not actually the function this gene evolved to do. That's what it does when it doesn't work. That's like saying that sugar makes coffee bitter when it's not there. That's not the function of sugar, right? That's not why you buy sugar. So the function of BRCA1 is that it is a transcription factor which regulates the cell cycle when it works. So the causal function of genes, what they do when they work well, you find it from their wild type healthy expression. It's also relevant to comparison between species, and a few of you are doing evolutionary biology. Hello. But even for the others, today, as you know, more and more to know function, we compare between species. So we do it all the time for sequences. You go to Ensemble or any other database and the genomes are aligned and you see the orthologs and it tells you something to know that your gene you're studying in mouse is conserved with human and rat or not. Well, we also would like to know if gene expression is conserved or not. And for this, we need what is evolutionary relevant, which is the wild type healthy. And we provide a reference for biomedical studies. That's actually one of our biggest use cases. If we look at our citations, people use a lot. They have a treatment, they have a mutant, they have cancers, and they want to know what happens when this is wild type healthy, and we provide this. So someone mentioned GTech. GTech is a very large project of gene expression to understand the relation between DNA level polymorphism and gene expression variation, so which SNPs guide change in gene expression. And in the version 6, here you have samples were collected from 54 non-diseased tissue sites across nearly 1,000 individuals. That's what's said first in the GTech. But if you go to the details of the documentation here, in the FAQ, which is very long, we have not excluded specific donors from specific tissues based on their cause of death or medical history. So in fact, here they're telling you it is not healthy. They do not guarantee this. So we recurated GTech data, and we found that we had many individuals that we could not keep in Bgee because they died of drug abuse, for example, or of cancer, or they had a BMI which is considered by the World Health Organization as unhealthy. And also among those who did not die of a cause which we consider makes the whole individual not wild type healthy, for example, drug abuse, I think you can say someone who died of drug abuse was not in the physiologically healthy state. Some had issues which make us exclude a specific organ. For example, if someone died with dementia or Alzheimer, we will not keep the brain tissues in Bgee. So in the end, we kept only half of all samples from GTEC, and another proportion, quite large, we kept no sample from individuals who died, for example, of drug abuse or cancer, and another fifth, we discarded some samples but kept other samples from the individual. So this shows the importance of curation. So we reviewed 12,000 libraries. Our curators read all the pathology reports, and we kept about half. And from this, we have all the information of the private annotations, which are partly confidential for respect of the privacy of the people who gave the data. So we make public from this the anatomical entity, obviously, so the organ, not the exact age, but an age range, so a destiny, the sex, and we cannot provide the ethnicity. So in the end, from all this work, we get 539 conditions, 75 anatomical entities, so organs, tissues, cell types. And you have a specific page here, the link, we'll share the slides afterwards, where you can find all the information on the GTEC data curated in Bgee. Another example of curation we did for wild type healthy and for the annotation is the fly cell atlas. So this is also a big project by a consortium which did a lot of their own annotation in-house. But when we re-annotated, we found many small errors not following standards. For example, the male reproductive system was reported with two different identifiers, so you couldn't get automatically all the expression from male reproductive system because it was put into two different boxes, so we put it together. Sometimes there was inconsistencies. For example, they annotated ovary cell in males or testis in females, which in Drosophila in the lab does not happen. And there were tissue annotation in the cell type column, so you're looking for a tissue and you don't find it because you look for it in the tissue, but it's actually in the cell type. So added hindgut, which is an organ, was in the cell type. So we corrected all these, and so in the end we have 61 libraries, 1,500 conditions, and we described 151 cell types, which we recurated carefully to the right terms. And from this, we get 27 million processed expression values because with single cell, you get a lot of data. And again, there is a specific page where you can get all the information about this experiment and download all the curated, annotated libraries from the fly cell atlas in Bgee. And so we have this page where we have the data sets of interest, which we are going to expand in Bgee 16 because we have now re-annotated, for example, all the human cell atlas, the Allen brain atlas, and other data sets which probably some of you know of. And so, yeah, we are continuing to expand this.
There was a question in the chat.
Yeah. Yes.
Did you provide any feedback to Flybase about the problems you discover? So the Fly database, I guess maybe you refer to the GTEC dataset.
Maybe this was the fly cell atlas, maybe,
Yeah.
no? So can you answer this, in fact, Frédéric?
Yes, so actually we collaborate with a group that participated in the annotation of the fly cell atlas. So yeah, we updated together the annotation, and now they are reported back these updates to the fly cell atlas and also to cell by gene, where you can access and visualize the clustering and the cell type. So yeah, through this group collaborating with the fly cell atlas team, we did give this feedback back. But so I don't know if they updated the fly cell atlas website. I know that the cell by gene dataset is gonna be updated. I don't know where it is in the pipeline towards the fly cell atlas itself.
Thank you. So here another little so a bit more complicated question. We have this experiment. Shall we show you the full description here? We have a circadian time course of liver mRNA profile of wild type, BMAL1 liver knockout, preverb alpha beta liver double knockout, cry1, cry2 double knockout after 12 weeks of high-fat diet feeding ad libitum or time-restricted feeding. I will copy this text to the chat so you have it fully. Don't answer yet, but I'm going to restart this because you need some time to read it. So where's the Zoom chat? Chat. There. So considering this, which terms would you consider fit the description of healthy for healthy wild type? So which terms are wild type healthy in this? So this is the kind of thing our curators do. I took a real example of a data set that was curated and annotated in Bgee. So put yourself in the shoes of this curator. What would you keep? Which samples would you keep? Which would you not keep? This is a tougher question. Answers are coming in a bit slower. I gave you a bit more time. And seconds. Put some answers even if you're unsure. So obviously, I think you all agree that wild type fits the expression of wild type healthy. Most of you thought that circadian time course is also wild type healthy, and this is correct. So this is the kind of experimental manipulation which will actually do variation in gene expression because genes are expressed differently at different times of day and night, but we consider it's within wild type healthy, and so we'll include it. Of course, liver is an organ which we want to capture. There was a few of you thought that the different diet restrictions were okay, but only a few of you. So indeed, we will keep this. We consider this is part of the natural variation within nature. Animals are not always eating the same, so variation in diet, if it stays within a reasonable range, which would be physiologically normal for this animal, we keep it. And the knockouts, obviously, we do not keep. So I think you followed pretty well what is a wild type healthy.
There are also two questions in the document that I can answer.
Yes.
So, do you include information from cell lines? No, we do not consider that as healthy wild type normal. And did you retrieve the individual age of the subject in GTech? Yes, we did. We do have access to the information that are restricted access, but we cannot make it publicly available. We have to provide a 10-year age range. So that's the information you're going to retrieve from Bgee, but we did have access to the exact information to provide this age range.
Thank you. So curation, then we standardize the annotation. So how do we do this? So first, standards are important. Standards are sometimes a bit boring, but it's really important because it allows data to be reusable. Every year, millions of dollars or euros or other currencies are spent on generating data sets in biology. And if they're not reusable after the first publication, they can be lost and not reused. So we try to make this usable. And so standardized data is reusable data. And standards define the necessary metadata. The metadata, if you want, the data is the counts of gene expression or the sequence, and the metadata is everything else, what tells you what this data actually is, which species it comes from, which organ, which conditions, and so on. So this data can be, we recover it. If it's missing, we contact the authors, and then the complete files are available from Bgee. So you would have in Bgee, this is a screenshot from Bgee, you would get the text description and a fully curated data with all the metadata standardized. And for single cell, we follow the H5AD standard, which allows you to put it into your pipeline behind. And we provide documentation of what we put into this H5AD so that if you are not sure of it or you have some questions, what is an H5AD, you can recover it easily, as well as example code how to integrate this into your analysis in Python. And for single cell, there's actually a jungle of protocols. It seems that every second paper doing single cell invents a new protocol. So we have classified 32 single cell protocols as of last year. We've stopped running after all the new protocols because we've decided for now to focus on 4 protocols, which are the most widely used and standard. But we take both SmartSeq, which are full length, and Chromium, which is 3' end tags, and we take both single nuclei and single cell. And so it's very important for us to capture this because then the processing, the treatment, and the interpretation you can make of them are different. And then when we have all this information, we annotate the expression to ontologies. So what is an ontology? An ontology is 3 things. First, it's a list of terms so that we agree on which words we use for things. But if you just have a list of terms, that's a controlled vocabulary. Then you need definitions of the terms. So not only which words we use, but what do they mean. If you have a list of terms with definitions, you have a dictionary. And then we add relations between the terms, and then we have an ontology. What is a relation? It is the brain is in the head. So there's a relation between brain and head. Brain is in the head. And we can have different types of relations. You can immediately see that brain is in the head, but it's part of the nervous system, and it's a type of organ, right? So you have different types of relations, and so we capture all this, and this allows what we call automated reasoning. What is automated reasoning? If I say I want all genes expressed in the head, and I have genes which are annotated to the brain, because I know in the ontology that the brain is part of the head, I can recover these genes expressed in the brain. I don't have to write explicitly these genes are expressed in the head. They're expressed in the brain. I ask for the head, I get the brain. That's automated reasoning. So the example of ontology I think you all know is the gene ontology. So the gene ontology has all these things. It has terms, it has definitions. So here you have cell migration in hindbrain, which is a term, and it has relations to other terms, right? It is a hindbrain development. It is a cell migration. A hindbrain development is a brain development. It is also an anatomical structure development, which is a developmental process, and so on. And these terms then, which are standardized, can be reused. For example, here is the UniProt SwissProt page of the homeobox protein HOXB1A in zebrafish Danio. And if you go down in the page, you find these gene ontology terms with sources in gold means that it's manually curated, which come from publications. And here you recognize ZFIN, the database of zebrafish, which did the annotation. And so because we all do the annotation on standardized ontologies, ZFIN, which does it in their model organ database for zebrafish, can be put into UniProt SwissProt because they both use the gene ontology. So we annotate an expression. Expression happens in organs, tissues, and cells. So we need to describe anatomy. And for this, we use an ontology called Uberon, the Uber anatomy ontology. Uber, because it describes not the anatomy of one species, but of all animals. So it was made by integrating the ontology describing different animals and then expanding this. And so it's quite a complex ontology, which will allow you to say that, for example, liver is part of the abdomen, is part of the exocrine system, is part of the hepatobiliary system, is found only in vertebrates. So you also have these restrictions to know by taxon, is part of the endocrine system, and so on. And so you can this way describe the complexity of anatomy in animals with terms which some are general to all animals and some are restricted to some species. Some relations are general, some are restricted, and various types of relations such as something which develops from something else, something which is part of something else, and so on. And we also use the cell ontology, which is ontology which describes cell identity, and it's quite complex, as you see here, and it also interacts with uberon because, for example, a hepatic sinusoid cell, a Kupffer cell is a type of hepatic sinusoid cell which will be found in liver blood vessel, and so you have relations in the cell types and the anatomical structures. There's quite a few challenges annotating single cell expression data to ontologies, notably because the cell ontology is running behind our discovery of new cell types and the huge diversity of cell types which exist. And sometimes we are unable to distinguish very specific cell types which can be distinguished in single cells. So, for example, you have these different types of neuron in the fly, T4A, T4B, T5A, T5B, which in fact in the atlas, in the ontology will all be annotated to the same term. And so we keep also requesting new terms and having to have new terms made. But because we try to do this carefully, it takes more time to get the new terms than to generate the data. So another little woo clap. If I have a data set that I annotate, and which has, I'm going to show you the description, tell me how many uberon identifiers, so unique terms in the ontology of anatomy, I should have for this description. So this is the description. We have 6 organs and 10 species of mammals and birds. How many different uberon IDs should be used for anatomical annotation? I will put this again in the chat so that you see it. Chat. There. Vote. So 6 organs, 10 species. How many different uberon IDs? In experiment GSC 3352. So how many different ontology terms of anatomy do we need to describe the data in this experiment?
Hello, good morning.
Morning.
How are you?
Is this Patricia? Sorry, I'm not looking
Yes.
at the pictures. Okay. Well, we're in the middle of the course. Okay, so we have an even divide between 6 and 60. So these both have their logic. We have 6 organs and 10 species. And before we had uberon, when we had an anatomy of an ontology of anatomy, per species, we would have needed 60 identifiers of ontology, one for each species. And then it would have been very difficult to compare them. When we started Bgee, that's what we were doing. But today, because we have uberon, which was developed between people from the Gene Ontology Consortium and us and some other groups, we have common anatomical ontology terms which are shared among species. And so for these 6 organs and 10 species of mammals and birds, we need only 6 different uberon IDs. So it makes it much simpler to recover. If I want the liver expression from 10 different species, I have only one query, one identifier to use to recover it. Okay? So that's the part of the point of doing standardization.
And
So,
sorry,
yes?
looking for the end of the book club, but there was a question some minutes ago more about the genes we can target in our analysis. So sorry, a bit disconnected from the ontology, but
So we target all genes which are annotated in the genome.
and the question is that do we integrate information about long non-coding RNA, transposable elements or retrogenes, and it's about like the protocol of the library we integrate. So most of the libraries are poly-A protocol targeting mRNA, but we do have some protocols targeting microRNA or long non-coding RNA. So we don't have that in all species, I think. But yeah, for the most widely studied species, we do have alternative protocols targeting other genes that producing mRNAs.
And concerning transposons, we take in genomes, what is annotated as a gene to call expression on. So we map to genes. So if the transposon, if a gene in a transposon was annotated as a gene, we will map to it. But if it is not annotated as a gene, we will not map to it.
So you can go to the specific page of a gene and see whether we do have expression information for that gene or not.
So rapidly, what makes an ontology useful? An ontology is meant to be a common standard. So an ontology is useful if it's used by many resources. For example, the gene ontology I showed you is used in ZFIN, Flybase, UniProt. So UBRON is used by generally everyone who maps expression or other function genomics to anatomy, including of animals, including humans. So, for example, it's used in GTEC, it's used in the Human Atlas, in the Fly Atlas, and so on. A useful ontology covers a large domain of knowledge. The gene ontology is gene function. UBRON is animal anatomy. And there are tools leveraging it. So most of you have probably done a gene ontology enrichment. Today you'll see the tools we use, which are all based on the fact that the data are annotated to these ontologies. And so I said we annotate to ontologies. So what do we annotate to in the end? What type of information? First, anatomy and cell type, what I've been telling you about, but also developmental and life stage. So all the developmental stages and also the life stages afterwards. So sexual maturation, aging, and so on. And for this, we at Bgee develop the standard ontologies which describe development and aging, which are used by all the other resources. For example, if you go to GTEC, the aging they use for humans is ours. Sex, which is simple usually to capture, so depending on species, male, female, or male, female, hermaphroditic. And the strains when we can, so this is a bit more complicated. There's not one standard. We try to follow the same standards as UniProt. So for the different strains of lab mice, of lab flies, but also the different human populations and so on. So here's an example what annotation would look like. This is from publication Single-Cell Transcriptomic Atlas of the Human Retina with age-related macular degeneration. And so you see the cell type annotation. And so here you have what the author wrote, which is non-standard, but was used in their paper. And then how we captured this in the cell ontology and which terms it corresponds to in the cell ontology, which will make it comparable between species. The same for the anatomy, how they describe it and how we capture it in the ontology. And here you have the age, and here they gave exact years. So we capture it in an ontology of human aging, goes to the granularity of the specific age. So you will have 68-year-old become 68-year-old, which doesn't seem intuitively like a huge gain, but because this is part of ontology, if later you want to capture, I want all gene expression in cells of humans which are in the age range 25 to, no, this is 68, so post 65, so which we would consider aging humans, then you will capture this automatically. You won't have to type in all the years. And so this is summarizing what we just saw, that in Bgee we do data curation, data annotation. So I hope you see the difference. The curation is finding the right data sets which are wild type, healthy, follow the right protocols. And annotation is saying which protocols are used, capturing this and capturing all the metadata, annotating it to ontologies. And this is where we are right now. So this is the end of this part. So now if there are
So there are questions in the document already.
So I will re-show the videos. There, I see everyone's videos again. Hello.
So there is a question, do you species definition include macrobiomes? So I guess it's microorganism in relation to a host. I'll say if it is an animal, yes, we might include it. Also, we don't have any for now, but most likely it's often not animals, but parasites are animals, but we don't have any so far.
And yeah, just to add on this, we don't aim to capture the microbiome. So like human is the genes which are in the human genome. It's not the genes of the microbes which are in the human gut, for example.
That was maybe microbiome, yeah. Okay.
Yeah.
And another question is, how do you match stages in different animals based on what? So that's a tricky question. So we created ontologies describing the development of different animals. It's based on criteria such as sexual maturity, or in mammals, age of weaning. So it's like, yeah, and then we carefully look into in each species, what is the typical age of sexual maturity, what is the typical age of senescence, and so then we map to these different terms in the ontology. But we also have in the ontology the exact age that can be reported. So, but it is difficult to just categorize developmental stages for sure. So if you have other questions on this first part.